Hey everyone, here's our fourth installment of our notes for chapter 8. I'll apologize in advance for any banging and hammering going on. So last topic here for this round of notes is on meiosis. So if you remember at the start of our notes, we discussed that cell division occurs in two ways: cell reproduction, either via mitosis, shown down here, or meiosis. And if you remember, meiosis is the process which takes place for those organisms that reproduce sexually. So just get my pen working here. So meiosis, as I mentioned, is a form of cell reproduction that occurs in organisms that reproduce sexually, right, involving two parents. If we remember back to mitosis, mitosis is what we refer to as asexual reproduction. And so if you think about throughout the course, I've tried to give you little strategies or things to remember about how things are named. So if you remember back to the monosaccharides and disaccharides, their name ends in ose, right? Lactose, glucose, galactose, those are sugars. If you think about enzymes, enzymes end in ase. So catalase, dehydrogenase, maltase, lactase, like we have all these digestive enzymes, etc. So when you think about a word with a in front of it, asexual, it means lacking or without, right? So essentially, asexual reproduction is you've got one cell, so a parent cell, right? So we might call this the parent cell, and that parent cell divides, producing two new daughter cells, and that is the function of mitosis. And what is important to keep in mind here is we're going to talk about the chromosome number, and this is the fundamental difference. Both types of cell reproduction involve parent cells making new daughter cells, right? But in the case of mitosis, we start off, if we're just using us as an example, humans, we have 46 chromosomes in our cell. And through the process of mitosis, when this takes place, the cell is going to produce two new daughter cells, each with 46 chromosomes. All right, but that is the fundamental difference in meiosis. And if you think about meiosis is responsible for reproducing our sex cells, our sperm and our egg, and those will have a very different number of chromosomes. So remember, don't forget though that mitosis occurs in all multicellular organisms. So what we need to understand is if you're a multicellular organism that reproduces sexually, then both forms of cell reproduction occur. Meiosis occurs to produce your reproductive cells, sperm and egg in the case of most animals, and that's true in plants as well, pollen and egg. And as well, you have mitosis, which occurs to replace cells that wear out, replace cells that die, or just overall growth of the individual, right? So that includes, replacing cells, replacing cells that wear out, or simply growth and repair, right? So that's the strategy of mitosis. Now, when we get into meiosis, we want to make sure we understand there's quite a bit of terminology here. And so I'm going to just pause the video and write down some of these terms, and then you can start the video to get the definitions. Okay, so now you've restarted the video. So a few terminology here. Somatic cells, these are cells of your body that are basically non-germ cells, right? They're any cell in your body that's not a sperm or an egg, that's not a reproductive cell. And one of the important terms here is that all body cells are what we say diploid. They contain two sets of chromosomes. and those two sets came one from each parent, right? Your sex cells, or we may refer to them as reproductive cells or gametes, okay, so another important term, your sperm and your egg, these are haploid and they contain just one set of chromosomes. So when we talk about our chromosomes, if we're talking about us as humans, we have a 1N or haploid number equal N equals 23. So we have 23 chromosomes that represent the human genome. But of course, as a diploid cell, right, any cell of your body that is diploid, then the 2N number represents 46 because the cells of your body are made up of two sets of chromosomes, one from each parent, right? And what we want to understand is that chromosomes come in pairs, and we refer to these as homologous pairs. Homologous chromosomes are essentially, so homologous chromosomes are two identical chromosomes or chromosomes representing the same DNA from each parent. Chromosomes that contain the same genes but have come from different parents. All right, so we have 23 pairs of chromosomes, and those pairs are homologous. They come in pairs representing genetic information from each parent. Maybe let's say this green one, to be stereotypical, is dad, and the purple one is mom. All right, and that's what chromosomes tend to look like, these sort of thickened, condensed rods, remember, when DNA is all wrapped up. But remember that upon DNA replication, right, that occurs during S phase of the cell cycle, they take on this sort of X form, right? And so these sister chromatids that are attached, remember, at this centromere, right, these represent genetically identical DNA. Genetically identical, okay? But these themselves, the homologous chromosomes, are not genetically identical. Okay, and to kind of give some foreshadowing here, not genetically identical, as to where we're going with respect to genetics as we move into chapter 9. And remember that one of the goals here was to sort of start to understand where inheritance comes from. If we were to imagine that, say, this chromosome contains information about your blood type. Okay, so this chromosome from dad here contains information about your blood type. And because this chromosome from mom is homologous, it also contains information about that blood type, but the information might be different. So whereas your dad's might have information for type A blood, your mom's could have information for blood type, but it might be different. Maybe it's type B, all right? When the sister chromatids are formed, whatever information here for blood type, whatever that original information on the parent strand of your dad's DNA, it would be copied to give a sister chromatid with that exact information. And similarly, if mom had information for blood type B, when her sister chromatid is copied and formed, that one would be exactly the same. And every single gene on those sister chromatids are genetically identical, but the homologous pair is not. And so here shows different colors and different bands which represent regions of a gene, right? And we might refer to these, I should have drawn my arrow up here. So these different positions of genes, these are what we call loci. And I think I've got it down here, locus or loci for plural. Okay, so a locus is a particular location of a specific gene on a given chromosome. All right, and yeah, this specific genes, like say, for example, blood type, wherever the locus is on your dad's chromosome is going to be in the same position, the same locus on mom's chromosome. Genes exist in the same positions, and we call that the locus. Or if you're talking about plural, multiple genes, we say loci or plural form, right? Locus one, plural loci. Now I mentioned up above haploid versus diploid. So when we're talking about this refers to chromosome number. All right, and so the term haploid means you have one full set. We say 1N, one full set of chromosomes. And this is chromosomes, sorry. This is typical for our gametes. Gametes are all haploid, and diploid means that you have two full sets. All right, and that would two full sets of chromosomes, and we would be referring to our somatic cells or our body cells, right? And so in the case of us as humans, our haploid number is n equals 23, and our diploid number is, if n equals 23, then 2 times n is 46, okay? And the little image shown to the right is what we call a karyotype. And a karyotype is just simply, karyotype is an individual's collection of chromosomes. And it's also kind of a verb in the sense that karyotyping, I'm spelling this wrong, karyotyping is a lab technique used by geneticists to produce an image of one's chromosomes. Much like the one shown up above here, to look for abnormalities or to look for abnormalities in one's chromosome or genetic makeup. And we'll talk a little bit more about that later on. Okay, so before we get into going through the process of meiosis, I want you to make sure that we understand the fundamental difference, right? So where mitosis is occurring from the moment of conception, right? So we have sperm fertilizes egg, right? And so if we think of this sperm is 1N, this egg is 1N. They fuse through fertilization, forming a single cell, which I referred to before as a zygote, which is now diploid. And through mitosis, we end up here as individuals. And meiosis really doesn't kick in until puberty, in which individuals start producing reproductive cells, sperm and egg. And it essentially, we'll look at it in more detail when we get to the reproductive system, but essentially it's a result of increased hormonal production that leads to meiosis being triggered within developing teens, right? And so the two big hormones are referred to as follicle stimulating hormone. So this is a hormone that increases in your body as you reach puberty, and it stimulates meiosis occurring in your reproductive organs. So it stimulates ovaries to start having eggs mature and be released, and in males in the testes, causing the development and maturation of sperm. And the other one, the other hormone is luteinizing hormone, which plays a little bit bigger role in your secondary sex characteristics, making boys boys and girls girls. So essentially, germ cells stop mitosis. So germ cells are the specific cells that will give rise to our sperm and egg. But up until reproductive maturity, they grow and divide under mitosis, just like any other cell as organs grow, right? As the body grows and develops. But as these hormone levels rise, it causes these germ cells to stop mitosis and enter into meiosis, and the cell reproduction now produces sperm and egg instead of just genetically identical daughter cells. So down below is a look at the stages of meiosis. The nice thing here and overall is that the stages are the same. There's prophase, metaphase, anaphase, and telophase, and you can see those outlined here. The big fundamental difference is such that in meiosis, there are two rounds. So there's meiosis 1 and there's meiosis 2. And what I want you to focus on specifically is what is the fundamental result of each. In meiosis 1, the goal is separation of the homologous chromosomes. Notice how the chromosomes are paired up. I'm going to get you to draw your attention back to your mitosis diagrams and look at the fundamental difference, right? In mitosis, chromosomes tend to line up single file. But in meiosis 1, they line up in their pairs. And this is what ultimately causes a reduction in the chromosome number. So in this case, if it was a human cell and we had 46 chromosomes, they would be lined up as 23 pairs. And so when anaphase takes place, you would have 23 chromosomes going this way and 23 going this way. So these two daughter cells that are produced in round 1 is the first step in reducing the chromosome number. Now you might think then in meiosis 2 that the number gets cut in half again, but look at what's happening now. In the second round, now those sister chromatids are being separated. Remember the copied genetic chromatids that were produced up here during cell DNA replication, right, and producing the sister chromatid. Now those are going to separate. So it doesn't change the chromosome number. If we start here in the second round with 23 chromosomes, we're going to end up with 23 chromosomes, and these will either mature into eggs or sperm, right? And that's where I'm going to stop it for now, and then we'll tie things together in our next class. All right, enjoy your final day off.